Hi, today we are going to see about antigen processing and presentation. Two different types of immune cells we are having. One is B cells and another one is T cells. B cells is called as antibody bearing cells. It will recognize the antigens directly, whereas the antigen recognized by the T cells, it requires an processing of an antigen. And who is doing this processing? The macrophages is doing the processing, and for the presentation, the macrophages is relying upon MHC molecule. What do you mean by MHC molecule? Major histocompatibility complex. We are having two set of an MHC molecules, the class 1 MHC molecule and class 2 MHC molecule. So, the class 1 MHC molecule is associated with an peptides from an endogenous antigens, whereas class 2 MHC molecule associated with an peptides from an exogenous antigens. There are two presentation systems we are having. What are those things? Class 1 and class 2 MHC molecule presentation, and we are having two different types of processing systems where exactly the processing of an antigen is taking place is matters here. So here one will be takes place in cytosolic and another one will be takes place in endocytotoxic. Antigens is processed through two separate pathways. One is cytosolic degradation and another one is endocytic degradation. In a cytosolic degradation, the endogenous antigens will form an vesicle cytoplasmic proteasome complexes in the presence of a protein called as ubiquitin and ATP. There it will form an peptides. That peptides will be translocated towards the endoplasmic reticulum through the DAP protein particles. And here in an endoplasmic reticulum, the class 1 MHC molecule will be present. The interaction between the peptide and class 1 MHC molecule will be takes place in endoplasmic reticulum. From there, it will form in vesicles. That vesicles will be translocated towards the surface of a cell. Coming to the endocytic pathway, the exogenous antigens will be engulfed by the phagocytic particles. It will form in vesicles. That vesicles will be combined with an lysozyme enzymes. It will degrade all the antigens into an peptides. And this peptides will be combined with an class 2 MHC molecule. Then this complex will be translocated towards the surface of an surface of an cell. So here the class 1 MHC molecule and class 2 MHC molecule is giving us a clear idea about the antigen, whether it is an endoantigen or else exoantigen. If it is an endoantigen means it will be expressed through class 1 MHC molecule. If it is an exoantigen means it will be expressed through class 2 MHC molecules. Now we are going to see about the video presentation of antigen processing and presentation by the macrophages.
Let's first study the processing and presentation of intracellular or endogenous antigens. This is a cell and its components which will take part in antigen processing and presentation. This is the nucleus, rough endoplasmic reticulum and Golgi apparatus. To understand this pathway, let's take example of viruses. Here the cell infected by the virus is known as the target cell. Let's zoom in and see how these viral antigens are processed and presented. When a virus infects a cell, it uses the cellular machinery of the cell to synthesize viral proteins. This occurs in the cytoplasm of the target cell. These proteins are taken up and digested in a large barrel-shaped structure known as proteasome. Proteasomes are present in the cytoplasm of all cells and are made up of 12 to 15 protein subunits. By the term proteasome we understand that these structures have proteolytic activity and thus they are able to degrade cytosolic proteins into peptide fragments. These fragments are of about 8 to 15 amino acids long. So, viral proteins are digested by proteasome into peptide fragments. In the next step these peptides are transported to rough endoplasmic reticulum. This transportation occurs through a pore formed by a protein, known as transporters associated with antigen processing, abbreviated as TAP. TAP consists of two subunits TAP1 and TAP2. The alpha chain and beta chain of MHC class I molecule are synthesized and translocated into the endoplasmic reticulum. These chains are assembled together and a group of chaperone proteins bring this molecule near the tap site. So, once the peptides have entered the endoplasmic reticulum, they bind to the MHC class I molecule in the peptide binding groove. Next, the MHC class I peptide complex thus formed is released from the chaperones and moves to the cell surface via Golgi apparatus. Once it reaches the cell surface, it integrates into the membrane and can be recognized by the T cells. For recognition purpose, T cells possess T cell receptors which recognize the peptides presented by MHC molecules. It also has the coreceptor CD8, which recognizes the alpha-3 domain of MHC class I molecule. Thus, T cells interacts with both peptide and MHC molecule on the surface of the target cell. Since this pathway involves MHC class I molecules, it is also known as MHC1 antigen presentation pathway. Processing and presentation of extracellular or exogenous antigens. As the name indicates, extracellular or exogenous antigens are the antigens that originate from outside a host cell. For example, extracellular bacteria. These antigens are taken inside by cells either by endocytosis or phagocytosis. The cells involved in this pathway are antigen-presenting cells. Recall that antigen-presenting cells are phagocytic cells which present peptides derived from internalized pathogens. These include B cells, macrophages, and dendritic cells. The pathway starts when the extracellular antigens such as extracellular bacteria are internalized by the antigen-presenting cells. Let's zoom in, to study this pathway. When an antigen-presenting cell internalizes a pathogen, it internalizes it into an intracellular vesicle known as endosome. In the next step, this endosome fuses with lysosome to form endolysosome. In the endolysosome, the internalized pathogens are digested by proteolytic lysosomal enzymes into small peptide fragments. MHC class II molecules are synthesized and assembled in rough endoplasmic reticulum. Here, the alpha and beta chains of the molecules associate with a third protein chain, called the invariant chain. This invariant chain binds to the groove of MHC class II molecule. This binding stops self-peptides in the rough endoplasmic reticulum from binding to the MHC molecule. These MHC2 invariant chain complexes are transported from the endoplasmic reticulum to Golgi apparatus and from Golgi apparatus to another vesicle. The invariant chain is digested and only a small fragment remain bound to the peptide binding groove of MHC2 molecule. This fragment is known as CLIP. CLIP stands for class II associated invariant chain polypeptide. In the next step, this vesicle containing MHC2 molecule bound to the clip fuses with the vesicle containing peptide fragments of internalized antigen. The peptide fragments displace the clip and bind to the peptide binding groove. These peptides are 11 to 15 amino acids long. The newly formed MHC2 peptide complex is transported to cell surface. Once on the cell surface, they are presented to T cells. These T cells recognize the peptide displayed by MHC2 molecule by T cell receptor. Another receptor, known as CD4 coreceptor, binds to the beta-2 domain of the MHC2 molecule. Since this pathway involves MHC class II molecules, it is also known as MHC2 antigen presentation pathway.